Students, up till now we see the breeding of fishes by the natural method, then wet bath and dry bath. These are also included in the natural breeding. But today we see the breeding, that is the induced breeding by hypophysation. Induced breeding means we have to induce the fish for the breeding. And here is the first method, that is the hypophysation. Now, what is this hypophysation? Hypophysation is nothing but the injection of the gonadotropic hormones into the fish body. Why? Because it is well known that the gonadotropic hormones, that is FSH and LH, secreted by the pituitary gland, play an important role in the maturation of the gonads and spawning in fishes. In this case, the Brazilians were the first to succeed in the inducing spawning by injecting fish with the pituitary extract. Then Russians also have successfully applied this technique for artificial breeding of sturgeons. Then in USA also they succeed in this experiment. But in India, Khan in 1938 was the first to successfully induce the Syrinus nugula to spawn by injecting mammalian pituitary hormone, and then Chowdhury in 1955 succeeded in inducing the Dandricus to spawn by the intraperitoneal injection of the pituitary gland from the cutla. So in this way, the hypophysation means to inject the hormones outside the body. As we know, naturally it is appeared in the fish body, but if we want to induce the fish before their maturation period, then we have to inject these hormones. When excess of these hormones get inside the body, the gonads get induced and start the spawning the eggs and the sperms. Now, in this case, what we have to do, the pituitary gland, we have to remove the pituitary gland and then they mix with the absolute alcohol in seal tube and desiccated at room temperature. Then acetone, which dehydrates and hardens the gland. After 36 hours, the gland is dried on the filter paper and stored in sealed vials in a refrigerator. The gland retains its potency for 6 months in acetone method and for 2 to 5 years in alcohol method. Then glands from freshly killed fishes are preferred, but those from 5 to 7 days old ice preserved specimen also gives good results. So here now we know that by injecting this pituitary extract inside the fish body, they triggered the fish gonads to produce the sperms and ova very early, naturally. Now here actually the female is generally given a stimulating dose of 2 to 3 milligram of pituitary per kg body weight of the fish. And again after interval of 6 hours, the female is given second higher dose of 5 to 8 milligram per kg of body weight. And in males are given a low dose of 2 to 3 milligram per kg body weight, and both are injected males and one female are now introduced into the cloth happa fixed to bamboo poles in a pond. So after injecting this hormone, we have to keep the brooder fish in a cloth happa in a pond. Then they get matured and they spawn, they lay the eggs and the milk is there outside the body of the fish and the fertilization is taking place. And after this, we have to remove the fish from the cloth happa. So here, this fish hormone, that is the pituitary extract, plays a major role in inducing the fish. Now, in the same chain, we have to see the next method of induced breeding, that is called as Chinese method. Chinese method is a very simple method, but here in Chinese method we have to create the situation, the natural situation that fish feels like this is the natural flowing of water, and because of that natural situation, the fishes get induced and the spawn is there. Now on screen, you can see one diagram of the Chinese hatcheries, their water supply and the hatcheries. So in Chinese hatcheries, this is the central tank, water supply tank. Here is we have to put the fish injected with the pituitary extract, and we have to wait for the some time. Now in this case, I am showing another diagram for the same thing. On the screen, you can see the tank. This is the main tank. See here, this is the main tank. This is made up of brick wall, and you see at the below there is a outlet of the water, and it is covered by a mesh. On the upper side, we have to keep the brooder fish, male and female. Now again, up on upper side of the fish, you can see a circular pipe and their outlet of the water. Now what happened in this case? The fish very get induced by the natural environment. So natural environment means what? So they need the natural flow of water, somewhat muddy water, lightning and thundering. So all these situation get induced the fish for the spawning. So that situation we have to create in the Chinese hatchery. So how it is created? See this diagram. When I see on the upper side the Chinese hatchery, you can see a small pipes are coming out. Now from the pipes the oxygenated water is coming out forcefully. So because of that the circular motion is created. So that circular motion of water is there and fish feels that this is the natural flowing water. So this is the main purpose to create the natural flowing of water situation in the Chinese hatchery. Now, after that, what happened? The fish feel that the natural flowing water, they spawn. The eggs are coming out, milk is coming out, and we have to then collected these eggs or seed on this mesh. Then we have to remove the fish, and the eggs are collected and they get flow inside this small hatchery. So this is the hatchery, and we have to keep the eggs there. They get fertilized and the small flies are coming out, and then that flies we have to transfer in the nursery pond. So in this way we have to induce the fish for the breeding in the Chinese hatchery. Now next we see the use of natural and synthetic hormones in the induced breeding. Now, in the use of natural and synthetic hormones in induced breeding, up till now we have seen the pituitary extract, then Chinese hatchery. In the same continuation, the hormone from that is the chorionic gonadotropin, then chorionic gonadotropin SCG, CG, and a pregnant mare serum. So, if we introduce these hormones inside the fish body, the fish get stimulated or the gonads become stimulated and they start the spawning. Besides this, certain gonadal steroids are also used for ovulating the fish. Now, in the same way, nowadays, the farmers use the synthetic hormones. For example, ovaprine. The ovaprine is the synthetic hormone